Okay, I've got a few announcements to make today. I won't be here on Friday, and there's a good reason for that. We've been given $1 million from the Howard Hughes Medical Institute to develop a program where we renovate the Biology 1A labs to kind of introduce new things like sequencing your own personal DNA. And there's a team of us that are working on that. That team consists of me, Mike Meehan, Jacob Mayfield, and this summer we'll be hiring 5 or 6 of you guys to work with us over the summer to develop these new modules. Now, as a result of this particular grant, though, 2 of us, Mike Meehan and I, have to go to the Howard Hughes headquarters for this week, and we have to fly on Friday, so we won't be able to be here. But you're in luck, because Friday's lecture will be given to you by Dr. Jacob Mayfield, who's also leading one of the lab sections for this class. Dr. Mayfield is one of the most impressively broadly trained biologists of his age that I know. When he got his PhD in the University of Chicago working on Arabidopsis, he discovered very important things about how pollen cells interact with flowers to lead to fertilization. Here at Berkeley, he's made really remarkable discoveries. He has found genes in mammals that help protect mammals and allow them to survive against the infection of what would otherwise be lethal fungal pathogens. So you can talk to him about that. And what he'll be doing is following on the themes that we've talked about today and trying to show you about the regulatory modules behind mitotic cell division, whereas today we'll talk about largely the machinery of mitotic cell division. So be here on Friday. It'll be a real treat. I'll return on Monday when we'll talk about meiosis. Now, to put the discussion into context today, I want to start by showing you this very short film by Julie Erringer that I think is really impressive. The image that you're looking at right now is the early embryogenesis of the small nematode worm, Sianorhabditis elegans, or C. elegans. You're looking at it after about 3 rounds of cell division. There's 8 cells there. And those big divot, clear, round areas are the nuclei. What I'm going to do is to start this movie at the place where some politicians would argue that life begins, at the moment of fertilization. Now, of course, that's an utterly stupid position, but the politicians hold it anyway, because, of course, the egg was alive, the sperm was alive before fertilization, so when life begins was something that was well resolved about several billion years ago. But nevertheless, we'll look early on and look at the events. Now, we'll show you the first stage, which will have 2 nuclei in the same embryo. Then we'll watch those nuclei fuse. And then we'll watch the amazing gyrations that goes through as the cell divides 3 times. You'll see those clear areas kind of disappear. You'll see a fibrous network appearing. Then that'll disappear. And then you'll see these clear areas. And then we'll go through the cycle again. So here we go. Right? OK, so there's the fused cells. The nuclei are fusing. Bingo! There's the first mitotic cell division. Nuclei are reforming. Nuclei are about to dissolve to prepare for the next round of cell division. There they go. Bingo. Look at that. Isn't that amazing? So this is the living image that I'll be showing you static steps of today. Now, this process can be very, very fast in C. elegans, or really quite slow in other cells. Want to see that again? Kind of hard to get it all the first time through. So there's 8 cells. Nuclei dissolved and now reforming. There's 2. Those nuclei are going to dissolve. Spindle sets up, separates. There's 2. Look at all the motion. Neat, isn't it? Okay. So we'll try to understand the mechanics that are going on in here, just in the course of our discussion today. A couple other points. There was some confusion between my schedule and what was on the website for my office hours, and I apologize for that. I'll have to straighten that out because I'm not going to be able to make that office hour that's on the website today. So I'll give extra office hours next week to make up for that, and my apologies. But I do want to bring up a couple of issues. I sometimes get asked 2 classes of questions that I don't answer. One class of question is, can you go over this topic or that topic from lecture? And the answer is no. And it's not because I want to be a jerk. What I'm going to insist upon is that you try to frame your question more precisely, because that'll help you understand what it is that you don't understand. And I'll ask any kinds of questions like that. Now, the other kind of question that I don't answer is, do we have to know this or that? Okay? Well, I'll answer it once right now. No, you don't have to know that. There are many careers available to you where the knowledge of that is utterly unimportant. I'll show you one. There's this gentleman couldn't answer most of the questions that you're answering me, and he's had a very high-profile career. Okay? So let's just talk about a few other things. Some of you want to know, well, why do I tell you all these stories, all this history? And the reason is that the context is really, really important. Okay. Anybody recognize this crazy picture? Yeah. What is it? Guernica, exactly. So you could look at this and say, oh, that's Picasso on LSD again or whatever, drawing these crazy things. And you don't understand the significance of that photograph unless you understand the context. And the context is on this slide. The guy on your left, Hitler, and the guy on your right, Franco, conspired to destroy the town of Guernica because that was where the opposition to the fascists were. And Picasso's famous painting is his reflection upon that tragedy. So without knowing the history, you don't really understand the context, and you can't really appreciate the beauty. So I'll show you some more slides on this later today as we talk about some of the principles during the class. So we can have that off now for a while. And what I want to talk about today is eukaryotic cell division, a kind of cell division that you should all be very, very interested in because it's critical for your life. And my hockey stick, or lacrosse stick pointer here, the eukaryotic cell cycle really has two phases of it that we have to be willing to consider. The phase that I'll talk about a lot today is the machinery of the eukaryotic cell cycle. And on Friday, Jake will tell you a lot about the control system and how that works. Okay? So the machinery is a transitory apparatus that assembles and disassembles and is remade every cell cycle. And as you will see, it is a really complex apparatus that has the capability of self-assembly, but its assembly is highly regulated in part by the control system. This apparatus consists of the microtubule cytoskeletal components that assemble into a structure called the spindle. And the principal proteins that we'll be concerned with in the spindle is tubulin, which comes in various flavors, alpha, beta, and gamma. And it's the alpha-beta tubulin that's principally responsible for building the mitotic spindle. Now, at the end of the cell division, we divide the nucleus. There is a contractile ring that forms around the cell, just as you saw in this movie. And that contractile ring is necessary to pinch one cell with two completely divided nuclei into two cells. That's made of a ring of intermediate filaments and actin and probably myosin to provide the force for squeezing the membrane into two. Okay. Now, the control system consists of three classes of entities. One is a protein kinase called a cell division kinase, abbreviated CDK. Jake will tell you the stories about CDKs, including the importance of it in the Nobel Prizes that were awarded to those discoveries. The cell division kinase has a regulatory subunit called a cyclin. In fact, there's many cyclins. And they are regulatory subunits of the CDKs that control its activity. And the other critical concept here is the see how all that regulation works on Friday. Now, one of the key concepts here is that the cell growth and cell division really have to be integrated. Okay? So think about this in the following way. Let's imagine that cell division happens when a cell has reached 90% of its proper size. Okay? As a result of that, you would get two cells, both of which are smaller than they should be. If that happened again and again, the cell would go from 90% of its normal size to 81% of its normal size to 70% of its normal size. And after a while, the cell would get smaller and smaller and smaller and smaller and smaller and smaller. Itsy-bitsy little cells, right? You'd have mitotic oblivion. On the other hand, if division was delayed 10%, each cell division, by the time 10 cell divisions went around, you would have these big, lumbering cells unable to carry out their processes. So there is a very deep and sophisticated integration of cell growth and cell division that we're really only beginning to understand the nature of the complexity of that integration so far. Now, we'll spend a lot of time talking about the events in the nucleus, the chromosomes, the mitosis, and that sort of thing, but we must not lose sight of the cells. And here's the funny thing about the stuff, this organelles. By and large, organelles require organelles for their assembly. If you put all of the components of an organelle into a buffer, generally, they do not form more organelles. So what we have to do is to figure out a way to make sure that when cells divide, all the organelles that a cell needs get segregated to the two daughter cells, just like the DNA and the chromosomes have to get segregated to the two daughter cells. Now, there are several strategies for doing this. One of the strategies for abundant organelles is to simply win by excess. So, for example, peroxisomes. There's zillions, there can be, depending upon the growth conditions, zillions of peroxisomes in a cell. So any place you cleave that cell, the two daughter cells will both get about the same amount of peroxisomes, as long as you cleave it in the middle. So any cleavage plane works for things like that. Now, chloroplasts and mitochondria are more sophisticated. In some cells, there is perhaps a single mitochondria or a single chloroplast. And the chloroplasts and mitochondria are actually ancient captured microbes, prokaryotes, that were captured by eukaryotes early in evolution. And they actually retain their own mechanism for fission. So as the cell and the cytoplasm gets ready to divide, chloroplasts and mitochondria have their own way of dividing by fission, much like E. coli did. And there are motors, we'll say more about motors, that attach to the outsides of these organelles that help direct them into the two daughter cells. Then there's a third strategy that's employed by entities like the Golgi or the endoplasmic reticulum. When Mark was describing the structures of eukaryotic cells, you probably got the notion that the Golgi was this big membranous floppy thing, and the endoplasmic reticulum was one big set of anastomosing membranes all interconnected. So the way those divide is that as mitosis approaches, what they do is to break up into thousands and thousands of little vesicles, and then those vesicles get assorted by just the plane of cleavage, and then after the cells break apart, all those vesicles reassemble into the Golgi and the endoplasmic reticulum. So there's many, many different sophisticated events that are all choreographed, and we're only beginning to understand this control system, and Jake will talk about that. Any questions so far? Yes, sir. What's your name? Russ.
Is there any sort of device or something inside the cell that causes the ER and the Golgi to come back together?
Yeah, Russ is asking the question, is there a device that causes the Golgi and the ER to come back together? Yeah, that's a really good question. Vesicles are a very common entity, a transitional entity in cell biology. And there's mechanisms for creating vesicles that produce on the surface of vesicles, what are the names of the proteins? Synaptotagmins and another class of proteins that I'm forgetting about, whose activity is regulated. And when they're ready to come back together again, these two classes of proteins allow the two vesicles to dock like this and essentially melt into each other. Okay? Thanks. Anything else? Okay. Let's move on to the classical representation of the cell cycle. I'll tell you the general belief about the cell cycle, then I'll tell you some of my doubts about the validity of that general belief. Okay? So the cell cycle is commonly divided into 4 phases. They've been called classically G1 for a gap and G2 for another gap to separate S phase from M phase. S phase is the phase called S for synthesis, during which the nuclear DNA gets replicated, the stuff we talked about in our last meeting. That's when DNA polymerase makes a second copy of all the chromosomes. Now, an important point is that the organellar DNA, that is the chromosomes in the chloroplasts or the mitochondria, are not controlled in the same way. Those mitochondrial DNA and chloroplast chromosomes are getting replicated throughout the cell cycle, not just in S. This is just referring to the nuclear DNA replication. M, of course, is the phase in the cell cycle named for mitosis. We'll talk about mitosis in quite detail. And this concept that these are gaps, I think, are really very misconceived. They were originally called gaps because when cells were looked at in this part of the cell cycle, people couldn't see what was happening. But in fact, I think of G1 as the executive phase of the cell cycle. Cells want to arrest in G1, with the exception of a very few cells like the Purkinje cells. Most cells want to rest in G1. They're safe in G1. And so in G1, each cell cycle, the cell has to decide, based upon its nutritional environment and signals that it gets from the outside, do I want to initiate the cell cycle? And if it does, then it goes all the way through without stopping. And if it doesn't, it has the option of leaving the cycle and going to a G0, or resting state. Resting state is another misnomer. Most of your cells are in G0. Okay? They are not resting. They're metabolizing like crazy. They're burning ATP like the band, but they're not going through the cell cycle. So most cells, most of the time, are in G0. G2, we've recently recognized, is a period during which one of many classes of quality control events happen, and these are called checkpoints. G2 is the time at which two kinds of checkpoints occur. One kind of checkpoint is looking over the chromosomes to see whether or not every single base has been replicated. If the bases have not all been replicated, cells hold up here in G2 until replication is finished. If the DNA has been damaged, the cells hold up G2 until the damage has been fixed. Okay? And in fact, my hunch is that G2 probably includes the last little vestiges of S-phase replication for parts of the genome that are hard to replicate. The other thing that G2 measures is whether the cells are really ready for mitosis. And I'll explain more about that as we go on. Now, the length of the cell cycle varies a lot. And most of the variation in the length of the cell cycle occurs here by the duration of G1. So for example, in your early development, some cells were dividing at a frequency of less than once every 8 minutes. Pretty darn fast. Typically, an intestinal epithelial cell, a tissue that turns over relatively quickly, those cells are dividing about once a day, whereas a cell in your liver divides about once a year. Okay? So it's a huge dynamic range. And your neurons never divide. Okay? So a lot of variability and flexibility in the length of the cell cycle, influenced by a variety of signals, largely external signals, that you'll hear a lot about when Gary Firestone lectures in the last third of the class. Okay? Any questions on the cell cycle so far? Remember, these are eukaryotic cell cycles we're talking about. Prokaryotic cell cycle is just replicate the DNA and divide as fast as you possibly can. Okay? All right. So we're going to move on to chromosomes now. You all have an intuitive understanding about what a chromosome is, but I'm going to try to give you more of a structural understanding. A chromosome, simply put, in eukaryotes, is nothing more than one long DNA molecule and all of the proteins that are associated with it. That's a chromosome. Now, it's hard to appreciate exactly how long a chromosome is, so here's an analogy. If we took your chromosome number 1 and expanded it so that its diameter was the diameter of vermicelli, thin spaghetti noodle, it would reach from New York to Miami. Okay? That's how long these chromosomes are. But all of the proteins that associates with them help compact them into a more compact structure, and we'll talk about that as well. Chromosomes, of course, have lots of genes, typically 1,000 or so on each of your chromosomes. And at the ends, there's special structures called telomeres, one here and one here. Telomeres refer to the ends of the chromosomes and the proteins that associate with those ends, and they solve a very important problem for us. if you thought through the issues involving lagging strand synthesis last time, you would have come up with a real problem, a conceptual problem. You would have discovered that there was no way to synthesize the end of the chromosome on the lagging strand, because there was no way to make a primer for synthesis of that last piece of the lagging strand. Therefore, you would have worried that chromosomes would get shorter a little bit each cell division. Well, they don't. And the reason that they don't is that there's a specialized enzyme called telomerase, which actually synthesizes extensions onto chromosomes in a way that is not templated by DNA. This was an enzyme discovered by a Berkeley student, Carol Greider, when she was working in Liz Blackburn's lab in Stanley Hall some years ago. Chromosomes also have a site called a centromere. This is a common point of confusion. Is it a structure or is it a site? It is a site on the, called a centromere, at which place chromosomes attach to the mitotic spindle. Okay. Now, at the centromere, at this site, a structure is formed, a very complex structure that we'll talk about, called the kinetochore. But the site is the centromere, and the structure that assembles on that site is called the kinetochore. So try to keep that straight. And of course, along the chromosome, there's typically on eukaryotic chromosomes, there's many different positions that each act as origins of replication. You recall that in bacterial chromosomes, there's one and only one origin of replication. In all eukaryotic chromosomes that I know of, there's multiple origins of replication. Okay? So what we're going to try to do now is to take this cartoon version of a chromosome and step our way, first steps into genetics. Any questions? Okay. Yes, what's your name? Miriam. It synthesizes extensions onto the ends of chromosomes. Okay. GGGGTT, GGGGTT, GGGGTT in humans. It just keeps putting that sequence again and again on the ends of chromosomes. Okay. All right. So there's two parts to genetics. One part is how, when one cell divides, we get two cells that are identical. The other part is how, when two organisms reproduce, we can get offspring which are different. What we'll talk about today is the first part. How is it that when two cells divide, we get two identical cells? And the answer comes in two ways. The first part of the answer is the fidelity of DNA replication. And the second part is the fidelity of chromosome segregation. You need both of those to create two identical cells from one. So again, bacteria have one chromosome that's typically circular, and eukaryotes, higher cells like us, typically have multiple linear chromosomes. I learned last year of the only exception to this I know. Apparently there is an exotic ant in Western Australia that has an entire genome of one chromosome, linear, but it's the only case that I know. In fact, I know of no organism that has two chromosomes, but I do know of organisms that have three different chromosomes, and I'll tell you about those. Now, eukaryotes have cells that can be in either the haploid state or the diploid state. And the haploid state is that in which there's one set of chromosomes, one copy of each of the different chromosomes. Okay. And of course, the diploid state, it has two sets of chromosomes. Now, if I were to ask you, are you a haploid or a diploid, you should say that's a trick question, right? Because you're not uniquely either. Your somatic cells of your body are, of course, all diploid cells, but the gametes used in reproduction are, of course, haploid. And the fusion of two haploid sets gives us the diploid. Now, there's something odd about the number of chromosomes that individual organisms have, which I think means that it can be anything and there's no really guiding principles. For example, there is a yeast called Schizosaccharomyces pombe that is haploid and that has three chromosomes. All of its genome is in three chromosomes. Its genome is about 16 million base pairs. There is a type of deer called the Indian monkjack that is diploid. It has a genome of about 3 billion base pairs, and it too has 3 chromosomes, though it is a diploid and hence has 3 pairs of chromosomes. So there is no relationship between the number of chromosomes and the sophistication of the organism. In fact, I believe the potato has more chromosomes than we do, so we should have a certain amount of humility about chromosome number. So N refers to the number of chromosomes. For Pombi, N is 3. For us, we have 23 pairs of chromosomes. Okay? The other issue that we need to concern ourselves, well, so how do you figure out how many chromosomes an individual, a species has? Anybody got an idea? How would you do that? What tool would you use? Pardon me? Chromatography? That's probably one of the methods that wouldn't work very well for measuring the number of chromosomes. What did you see in your textbook? Did you see chromosomes in any of the illustrations? Yeah, microscopes. Stain cells with something that stains DNA. You look in the microscope and you can count the number of chromosomes. Very straightforward. However, there's another tool that we need, which is the ability to count the amount of DNA in a cell. The amount of DNA in a cell is referred to as the C value, so that's DNA content. And the C value does not refer to the amount of DNA in base pairs. It refers to the relative amount of DNA depending upon where the cell is in the cell cycle. So 2C is the number given to the amount of DNA in a diploid nucleus when a cell is in G1. And of course, in G2, after S phase, the amount of DNA that is in that nucleus would go up, and so that would be 4C. And values between 2C and 4C is the value of the DNA content in a cell between S phase and M, between G1 and G2. Okay? So how do we measure this? Well, the way we measure it involves a device invented at Stanford by the Herzenbergs. Let's imagine a whole population of cells of which I've illustrated only one. We're going to treat those cells with a fluorescent dye, and the dye has 2 qualities. It only binds DNA, and it binds all the DNA equally, so you can saturate the amount of DNA. Therefore, the amount of bound dye is directly proportional to the amount of DNA in the cell. One then washes out the excess fluorescent dye, and you drip a stream of cells through a device called the fluorescence activated cell sorter, abbreviated FACS, F-A-C-S. So what this consists of is a very fine nozzle through which cells can go, and a laser interrogates the DNA content of each cell by illuminating that cell with the wavelength that is absorbed by that fluorescent dye. And if the dye absorbs that wavelength, it then emits photons of a different wavelength, and we have a detector and an amplifier that goes to a computer that keeps track of each cell that goes by. And then, if we're interested, we can actually use that computer to control the charge on a pair of chargeable plates by which the cells pass. Now, the surfaces of cells are typically negatively charged under normal conditions, and by adjusting the charge distribution on these plates, we can deflect cells to the left or to the right and into one tube or another. So if we use fluorescent dye that binds DNA, we can use the fax machine to sort cells that are in G1, that are 2C, from cells that are in G2, 4C. Okay. Or if we don't want to sort, but we just want to analyze the population of cells, we just run them through here and let the computer keep track. Okay. Now what's really impressive is the efficiency of these instruments. These instruments can analyze about 1 million cells in a minute with very high precision and give us a nice profile of what the population of cells is doing with respect to the cell cycle. It's also extremely versatile in studies of the immune system, and Gary Firestone will talk a bit about that, because immune cells have many different surface proteins that we want to know about and characterize, like the famous CD4 receptor for HIV. And you can use an antibody that's fluorescent instead of an antibody, a fluorescent dye, to react with a population of cells and sort out those cells from all the other cells that happen to have the protein recognized by that fluorescent antibody. So it's a very useful instrument. Now, what I want to concentrate on, though, is to interpret the data from such an instrument. The computer will print out a graph for us in which the y-axis refers to the number of cells that have gone by, and the x-axis is the relative fluorescence of each cell. So this curve will represent the distribution of, with a minute's processing time, about 1 million cells. So this many cells have 2C DNA content, and this many cells have 4C DNA content. Okay? So where in the cell cycle are the cells under this peak? G1, right? Over here, G2 and M. What about these guys? Where in the cell cycle are cells from that part of the graph? Right through here, right? Is there anybody that doesn't understand that? All right. Now, here's what I'd like you to do. I'd like you to spend a minute and draw on your notepad what you think the profile would look like from a cell that has, let's see, how do I want to do this? I want to profile, I know, you guys on the west side of the auditorium. I want you to draw a profile of cells that are stopped in their tracks by a drug that stops DNA replication, okay? And I want you on the east side of the auditorium to draw for me the profile of cells that have stopped in their tracks because they've been hit with a drug that blocks microtubule formation. So take a minute and draw that out. Discuss it with your neighbor. And then we'll get a volunteer to come down and help us out. You got it? Anybody need more time? Need more time? Okay. I would like you to come up and help me out. Did you get it? Okay, come on up. What's your name? Ben. Ben, come on up. Ben, here's the chalk. I want you to show me what you came up with as the profile for a cell analogous to this, a plot of the number of cells relative to fluorescence of cells that were stopped in their tracks by an inhibitor of DNA replication. What do you think that would look like? Ben is impressing the bejesus out of me. He's labeling the axes on his graph. Wow, that's good form. Did everybody come up with that? That's a darn good interpretation, Ben. And whether the peak is exactly on 2C or someplace between 2C and 4C depends upon the potency of the drug, right? But one peak no more than, not at 4C and no less than 2C. Good job. Okay, let's see if the East can produce an Eastern version of Ben. Would you like to come up and help us out? Yeah, you. I know it's no panic, but that's okay. So come on. What's your name? Yeah, you. Melton? Elton, come on up, Elton. Okay. So now on Ben's graph, I want you to show me what you would think with a dashed line so that we can distinguish your graph from his about what it would look like if we did a profile on a population of cells treated with an anti-cancer drug, for example, that blocks cells in mitosis by depolymerizing microtubules. Okay? Everybody agree with Elton? Pretty good. Elton, I would quibble a little bit in that I wouldn't want to see a curve that goes up at these intermediate values between 2C and 4C, but that's sort of artistic license, right? Okay, good job. All right, excellent. You guys have that point down. It's really important that you do because it's astonishing how often that kind of a problem shows up on exams. Right? You don't have to remember anything, but you have to figure something out. So now, for purposes of illustration, I want to consider mitosis in the somatic cells of our friend, the Indian monkjack. Now, I told you the Indian monkjack had 3 different chromosomes. So there's 3 pairs of chromosomes. One of each of these it gets from the mother, and one of each of these it gets from the father. Right? So what I'm showing you here is a representation of the nucleus in G1 of a cell from the Indian monkjack. So we have 2 blue chromosomes, 1 paternal and 1 maternal, 2 purple chromosomes, 1 maternal, 1 paternal, and 2 pink chromosomes, 1 maternal, 1 paternal, 1 whatever. Okay. So first question is, how many DNA molecules are there in this nucleus? 6. 1 DNA molecule per chromosome. A chromosome is 1 DNA molecule from one end to the other. Now, this pair of chromosomes goes by a special vocabulary word, which is homologous chromosomes. Homologous meaning about the same thing. Okay? I'm going to come back and put a more rigorous about definition on that in just a minute. They're nearly identical sequence. Now, in G1, there is a clear nuclear membrane and defined nucleus. But you saw in Julie Ehringer's movie that when the cell started to divide, the clear nucleus and its outline disappeared. And that's, in fact, what happens in eukaryotic mitosis. Early in mitosis, the nuclear envelope dissolves. What it really does is to break up into vesicles, just like the Golgi and the endoplasmic reticulum, and then reforms after mitosis. Those vesicles refuse. Okay. Now, M phase, early mitosis, is after S phase. And so, of course, during S phase, DNA replication has replicated all of our chromosomes. And now, what was a single blue line is a pair of blue lines that are referred to as sister chromatids. Each sister chromatid is identical in DNA sequence because they are the products of the DNA replication of one of the 2 homologous chromosomes. So sister chromatids have identical sequences. And these are pairs of homologous sister chromatids. So how many DNA molecules in the nucleus of monkjack, in that monkjack cell at that point? $12. Yeah, no problem, right? Now, the amount of condensation of these chromosomes, I told you before, the vermicelli that stretch from Miami to New York, is really quite impressive. In real length, the length of the DNA of a human chromosome, like chromosome number 1, is about 20 centimeters. When the chromosomes condense in early mitosis so that we can see them, they condense to about 2 microns. So that's a 100,000-fold shortening of the DNA molecule. And that happens because of interactions between the proteins associated with the DNA all the time, and other proteins called condensins, which condense chromosomes so that in mitosis they no longer look like very, very thin threads that are almost invisible in the microscope. They look more like something like this. So this would be a representation of a pair of sister chromatids. Okay. Now, this pair of sister chromatids is not only condensed, it is cohesed. Now, in some textbook illustrations, you will see pictures which make you think that chromosomes are held together at the centromere. That's not true. Sister chromatids are held together along their length by a proteinaceous glue that we know a lot about now called cohesin. The cohesin glue is put on chromosomes when they are replicated in S phase. But until they are condensed, we don't see how they are glued together. So it's the combination of condensation by condensins and cohesins that's responsible for these figures of which your text has a beautiful electron micrograph of. Now, I told you before that the centromere is the point at which a chromosome attaches to the spindle, but the structure that assembles at the chromosome that does the attachment is shown here as the kinetochore, and that's a very complex protein complex that I'll show you pictures of in just a second, and it does the job of attaching this point to the mitotic spindle. Now, what we've covered is pretty simple, pretty straightforward. Your text does an excellent job describing the details of mitosis, and I recommend that to you. But I found over the years that this has a tendency to confuse people. So let's stop and see if there's any questions about it. Or see if I've made a mistake anyplace. Yes, sir, what's your name? Alan, what's your question? You know, I've seen it both ways. Mike, how do you use it in the lab? That's a good way of doing it. So N for humans would be 23, number of different chromosomes, but 2N diploid N would be 46. Okay? Don't worry about it. I will make it unambiguous in any question that I ask you. Okay. Other questions? Yes, Marianne. Is it Marianne? Martha. Sorry.
What are the cohesins made of? Are they like protein, or are they, I don't know, something else?
Yeah, the question is, what are the cohesins made of? They are made of approximately 4 different proteins that have names that we know about. And there is a really active controversy right now about how the cohesins actually work. There's one school of thought that actually says that the cohesins are wrapped entirely around the DNA like a napkin ring and holding them together. There's strong evidence in favor of that. But Doug Koshland, my friend, has a different view, and hence I favor it. And Koshland's view is that these are rings of proteins that snap together along the face at which the chromosomes come together. All right? So there's a debate now between the pure glue model and the napkin ring model, and it's going to get resolved in the next year or two. In fact, I believe there's a seminar today at 4 o'clock by one of the people from the napkin ring school of thought coming to defend that view of how chromosomes are held together. Other questions? Okay, are we ready for the action? All right. Now, there is a point that is utterly simple, but you must understand about mitosis. And that point has to do with the disposition of chromosomes in metaphase. Now, you've heard about the metaphase plate. Do not think of this as something that you can eat dinner on. The metaphase plate is nothing more than a conceptual plane along the midline of a cell. And in metaphase, pairs of sister chromatids align along that conceptual midplane at random with respect to their homologous partners. Okay? So there is a pair of sister chromatids that are the homologous partners of that pair. They're not aligned near each other whatsoever. Right? So one pair of chromatids does not know what the other pair is doing. This will be in stark contrast to what we'll talk about in meiosis on Monday. And to avoid confusion for the rest of your life, all you got to do is understand the difference between metaphase of meiosis and metaphase of mitosis. So get this down now. Okay. Now, there are structures called centrioles which organize the growth of microtubules from them. And there's two classes of microtubules with respect to mitosis. I'll deal with one class now, and that is the microtubules that connect the centriole to the kinetochore on the centromere. Microtubules have a plus end and a minus end, which represents nothing more than the asymmetry in the individual tubulin molecule when they assemble into a polymer. So by convention, we call this the plus end and that the minus end. Now, once all of the chromosomes are attached to the spindle, but not until then, once they are attached, then the cell uses the regulatory machinery that I alluded to earlier to activate a protease which dissolves the cohesin that holds the sister chromatids together. At that point, the chromosomes migrate towards either pole, and the issue is what provides the force for their migration. Now, here at Berkeley, Eva Nogales has come up with a very new and different theory about how this happens. In the past, it has been thought that motors, protein motors, in the kinetochore actually drove the chromosomes along the microtubule like a car along a track. Eva has a very different view and some very strong evidence in favor of it. What Eva has shown is that the microtubule, like a tubule, at the ends, once all the chromosomes are attached, starts to peel apart like you would peel a banana. So we're going from a highly ordered state to a highly entropic state. And of course, you gain free energy as you increase entropy. And so Eva's view is that there is a destabilization event that happens here, and the peeling back of the end of the microtubule causes the chromosomes to slide along the retracting microtubule to the pole. Okay? I'll show you perhaps some electron microscopic evidence in favor of that. It's really quite remarkable. If I don't show it to you today, I'll show it to you Monday. So anaphase, then, is the part of the cell cycle in which the chromosomes go to the poles, perhaps driven by the depolymerization of the end of the microtubule. Now let's talk a little bit about microtubules so you understand this in greater point. Microtubules are tubulin monomers plus GTP, puts them in a state where they start to assemble sequentially, first into rows, then into sheets, two-dimensional sheets, which then self-assemble into microtubules with a minus end and a plus end. Now, the old view that motors move chromosomes came from the fact that there truly are motors. There's two kinds of motors in the world. There's minus end directed motors, motors that on this roadway always go to the minus end, and other motors that are called plus end directed motors that on the highway always go to the plus end. And there is, in fact, a minus end directed motor that is part of the kinetochore. Whether that's critical for the force to move chromosomes or not, I think is an open question at this point. But it is ambiguous. The positive end-directed motors play a very important role, because in addition to these microtubules that attach to kinetochores, there are other microtubules that extend from one centriole across the midline and cross like this. The part of the challenge of the mitotic spindle is to position itself in the middle of the cell so that when the cell divides, it divides in the middle. So as the cell gets bigger, the spindle has to get bigger. And the way that it keeps itself in the middle, in part, is by having near the end of a microtubule an attached motor that is directed towards the positive end of microtubules. So here's a motor attached to the positive end of this microtubule. When these two come in contact, that motor wants to move towards the end, the positive end of this microtubule. If it's attached here and it's tracking along there, the net effect is to push that microtubule that way and this microtubule that way. And in this way, the action of motors on opposing microtubules helps position the spindle in the center of cells, allowing for an orderly plane of cell division. Okay? So the remarkable thing really about mitosis is how much our view of the coordination of it is happening. You heard about it in high school. Your eyes were glazing over early today thinking this is all known. But in fact, it's not. It's a really very active area of research. There's at least 3 labs in MCB that are making very fundamental contributions to our understanding of the details of mitosis, and we'll tell you more about those as the semester goes on. I'll show you the remaining slides. Probably either Jake will show them to you on Friday or I'll show them to you next Monday. So be prepared for Friday. It'll be a lot of fun.